All right, in this introduction, we're gonna start microbial metabolism. Now, the word metabolism because usually you referred to it as people saying I have a fast metabolism or a slow metabolism, but the word metabolism just means the chemical processes, the equations, those chemical equations that happen in the cell. So the different things that are getting broken down, the different things that are getting built up, all of those different chemical equations that are happening in the cell, that's metabolism. So if someone has a fast metabolism, things are getting broken down in the cell very quickly, things are getting built very, very quickly, sometimes it's more efficient, but it's just all the chemical cells that are happening in all the chemical processes that are happening inside the cell. So in this PowerPoint, some of the things that we're going to cover is some metabolism basics, some of the chemical reactions, how do enzymes play a role? And that's what I'm going to try to get through in this recording. The carbohydrate metabolism, anabolism, and regulation, we'll try to get all done in person in lecture. So first, just starting out with some metabolism basics. Metabolism is, again, just a collection of all the chemical reactions that are happening inside a cell. And when we're talking about bacteria, which are only one cell, it's happening inside the microbe. Now, there are 8 elementary statements when we're talking about metabolism. And the ultimate goal for a bacteria to have all these chemical processes occurring, if you're a bacteria, your ultimate goal of all these chemical processes is to reproduce, so that 1 bacteria becomes 2 and 2 becomes 4. That's what's happening inside the cell. It's constantly just breaking things down, building things up so that it can grow and divide. Now, before I go through the 8 basic statements of metabolism, a couple groupings, because I've got that. Yep. A couple groupings of types of chemical reactions. It's like 2 types or 2 groups of chemical reactions that happen inside the cell are catabolic reactions, or catabolism, and anabolic reactions, or anabolism. Now, in any kind of catabolic reaction or catabolic pathway, we're taking large molecules, we're breaking them down into smaller molecules. And when you do that, when you take a large molecule and you break it down into small molecule, it's called an exergonic reaction, and energy is released. It's ultimately you're breaking bonds, releasing energy. So you're breaking things down so nutrients can come into a cell, it gets broken down. So instead of having, say, super really big proteins, we can break the bonds in the protein into more simpler amino acids, which are the building blocks of proteins. The opposite is an anabolic pathway, is we're taking small molecules, we're putting them together and building things up into larger molecules. So we can take those tiny little things like amino acids, rearrange them, assemble them, string them together, and then we can build larger different things like proteins. So we can eat one kind of protein, break it down, and then build it into another kind of protein that a cell might need. Now, when you're building things, it requires energy. That should not be releases. That should be requires. Sorry. It's an endergonic reaction. Ender meaning goes in. Energy has to go into the reaction for it to occur, which makes sense. If you're building something, if I'm going to build, a house, I'm going to need some energy to do that. Now, because reactions are opposite of each other, and one easy way for myself personally to remember what's happening in a catabolic reaction versus an anabolic reaction, if you have a cat, or if you've ever known anyone that has a cat, cats like to break things constantly. Like my cat's one of those cats that if there's something on the edge of a table, she will knock it over. So cats like to break things. So I like to think a catabolic reaction is constantly breaking things down. And then I just have to remember the anabolic reaction is the opposite of that. Now, in all of the breaking things down, building things up that are happening inside the cell, there are 8 elementary statements of just kind of the basics of what is happening, of the breaking down and building up. So the first one is that every cell acquires nutrients, and I'll list them all out on a separate slide coming up. You can pause and write them all down as well. But every cell acquires nutrients. A bacteria needs to eat. It can't do anything without getting nutrients going in first. The second is that metabolism requires energy. So all of these chemical reactions are requiring energy depending on the type of organism. It could get the energy from light, but most do not. So the energy is going to come from the catabolism of the nutrients, aka breaking it down. Because in a catabolic reaction, when you break things down, it does release energy. And some of that energy is lost as heat in the cell, and some of that energy can then be stored. So nutrients go in, they get broken down, energy is released, some of that energy is stored in ATP. The fourth statement is that the cells, as they break down those nutrients, not only do they get energy released, which can be stored as ATP, they're also getting some of those precursor molecules, the small building blocks that are then used to make whatever the cell needs. In 5, we're now going to take energy stored in ATP. We're going to take some of those building blocks, those precursor molecules, usually some enzymes as well, and we're now going to start to build things. In 6, as we're building things, as you start to assemble some of those things, we start to get things called macromolecules. So instead of just maybe making a couple amino acids strung together, well, now let's get even more amino acids strung together. Let's shape them a little bit, and now we can have larger molecules called macromolecules. All an anabolic pathway, all of this green line anabolic. We're building things up. Now, as it's building things up, the cell is making more things, it's making more structures. The ultimate goal is to make everything the cell needs. It needs to make more ribosomes, it needs to make more parts of the cell membrane so that ultimately it can grow and finally divide. So once they've doubled everything they need, they will split. So here I've got written down, again, the cell gets nutrients, it gets energy, sometimes from light depending on the organism, but otherwise it gets its energy from breaking down those nutrients. That energy can be stored in ATP. As well as breaking down nutrients and getting ATP, we get some of those precursor molecules. We start to put those precursor molecules together, plus the energy, usually with some enzymes, and we start building things up. We start building larger and larger things. Now we've got macromolecules. We start to assemble them all into things like organelles, and then they eventually double in size. So that's your basics idea of metabolism, what's happening in a cell. Now, we're going to focus specifically on bacteria cell, which ultimately then grow and divide, but our cells do very similar things. Our cells take in nutrients, break things down, build things up, whatever cell I need. It just doesn't grow and divide binary fission-wise. It's a whole nother mitosis process. Now, as you have all of these growing and dividing and molecules getting formed and molecules getting broken down, one of the other things that's happening as you are breaking things down and releasing energy that's stored as ATP, when you break a bond, and when we say it releases energy, a lot of that energy that's released is the energy stored in electrons. So as you break a bond, and I say, it releases energy, well, what does energy look like? It's generally losing of electrons. Electrons don't like to just hang out by themselves. They need to be tied to something else. So when electrons are moving around, they're really taking energy with them. So when we're talking about moving of electrons, as we're breaking bonds and going through these catabolic reactions, when we're specifically talking about electrons, we're talking about oxidation and reduction reactions. So what type of thing is losing an electron, and then what molecule is picking up an electron? So the thing that loses an electron is the donor. The thing that picks up the electron is the acceptor. And they always happen at the same time because electrons do not like to float off by themselves. So this is showing that my substance A is going to lose an electron, but my substance B is going to gain the electron. Now, when something loses an electron has a negative charge. So because A lost an electron, it becomes positively charged. When B gains an electron, negatively charged, it becomes negatively charged because it gained something that was negative. When something's losing an electron, like my A molecule here, that's called oxidation. When something gains an electron, it's called reduction. And this right here is what trips students up all the time, that something called reduction means gaining of an electron. Because usually when you hear the word reduction, you're thinking less of something. But the reason why it's called reduction when you gain an electron is because what you're gaining is something negatively charged. So if you're gaining something negative, you are becoming reduced. I always like, if you start to gain a lot of negative friends, you yourself are becoming more reduced or negative. So that's why it's called reduction, is that charge is becoming less and less because it's becoming more and more negative. So even though you're gaining something negative. Another way that I like to remember which one's gaining, which one's losing, is if you can remember the acronym oil rig, like where they go drill for oil. The word oil means oxidation is or involves loss of electrons, whereas rig, R-I-G, reduction involves gain of electrons. So oxidation involves loss, reduction involves gain. Oh, there's my acronym. I thought I had it on here. Now, as things are getting broken down, again, nutrients go into cells, things get broken down, electrons are getting lost, other things are picking them up. There are some molecules that are constantly patrolling looking to pick up electrons. They're like the little taxi cabs, the little Lyft drivers that are just constantly looking to pick up electrons. And again, electrons have energy. So when something picks up an electron, it's actually picked up not just the electron, it usually also has a hydrogen molecule with it, but it now is actually energized. It has more energy to it because it's got this electron. There are 3 important electron carriers that are found inside the cell. I don't need you to know the full written out word, but if you're curious to know what the letters or abbreviation stands for, I have them included on here. But I mostly, want you to recognize these 3 abbreviations and know that these are electron carriers. Once they have that H on it, that means they've picked up the electron and they've picked up with it a hydrogen ion that likes to tag along with the electron. So NAD plus when it picks up electrons is now NADH. NADP positive or plus when it picks up electrons is now NADPH. And FAD when it picks up electrons is FADH2. So anytime you see NADH, NADPH, FADH2, this means this is an electron carrier. It has electrons that's picked up, and because of that, it's picked up those electrons, it now has energy as well. But it's not the only place where we can have energy stored. So we've got electron carriers that are storing energy in the form of those electrons. Is when you have a catabolic pathway and you break something down, you break those bonds, and we already stated before, energy is released. Some of that energy can be stored in ATP. And where that energy is stored are in high energy bonds. So we can break bonds and release energy and then form bonds to store that energy. So this is showing what the whole molecule of ATP looks like, and it's called adenosine triphosphate because it has 3 phosphate groups. And it's these bonds that hold those 3 phosphates together is where we can store energy, because the bond that holds there is a very highly energized bond, which means if you break it, you release a lot of energy. So once we've eaten some food, the cell has eaten some food, it's broken it down and released energy by breaking bonds, we can take all that energy and store that energy in these high energy bonds just by adding phosphates on. By adding these phosphate groups on, it's called phosphorylation. So ATP, adenosine triphosphate, we've got lots of high energy bonds here. But if it was missing a phosphate group, it wouldn't be as energized, and it's called adenosine diphosphate because it now only has 2 phosphate groups. When you add a phosphate on to go to ADP to ATP, that's where you're now storing some energy in those bonds. And then an anabolic pathway that requires energy, super easy way to get it is you just break one of these phosphate groups off and it goes from ATP to ADP. Now we can gather the energy from that. And if you wanted to break off another phosphate group, you can. The cells can, and they can go from ADP, adenosine diphosphate, to AMP for adenosine monophosphate. Lots of energy in those bonds. Now, the last thing we're going to talk about are proteins and enzymes. So enzymes are proteins. So a little review of proteins. I put just a lot of pictures on here. Proteins are made up of amino acids. I thought this was funny. There are 20 different amino acids that can be found. Bacteria can have even up to 22. Human bodies have just 20. Amino acids are all structured the exact same. They all have a single carbon. They have a single boring hydrogen on one side. They have an amino group on one side, a carboxyl group on the other side. And the only thing that changes between those 20 or bacteria 22 different amino acids is that R group. Like this one just has a single H. This one's got this cool hexagon shape on it. 20 different amino acids, 22 for bacteria. When we put amino acids together, so here's one amino acid, here's one amino acid again, structure, similar. We can put them together and they're called peptides. This is called a dipeptide because there's just 2 amino acids together. But you can string thousands of amino acids together. And when you have this long chain of amino acids, it's now called a polypeptide, which just means lots of peptides or lots of amino acids. And once you take that amino acid, or that's chain of amino acids, that polypeptide, and you twist it and you shape it into whatever shape it needs, now it can actually do what it's supposed to do. And so let's talk a little bit about enzymes, proteins, and how they are now playing a role in all of this. So enzymes, they are proteins. So enzymes are proteins, but not all proteins are enzymes. But enzymes work as an organic catalyst, which means their job is to try to make sure that reactions happen. There are lots of reactions, catabolic reactions, anabolic reactions, and if enzymes weren't present, some of those reactions wouldn't occur. So if enzymes are present, they increase the likelihood of a reaction. How they do that is they lower the energy that's needed to start a reaction. For example, if these reactants want to become these products, so this double molecule AB wants to split up and become A and B. That's what it wants to do, but it can't spontaneously do that. It first needs a little bit of energy to start a reaction off. The energy that's needed to start any reaction off is called activation energy. What enzymes do is they lower how much energy is needed to get that reaction going. So here in the dark red line, it's showing how much energy is needed. So if we start with reactants, we have to put in this much energy to get their reaction started to make the products. But if enzymes are present, we only need this little bit of energy. And if you don't need as much energy to start the reaction off, it's more likely to happen. My example is if you had a matchstick and you looked at it, it's not just going to spontaneously light. You can't just look at it and say, okay, matchstick, light. You have to put a little bit of energy in. You have to start the chemical reaction. You have to add that energy. You have to add the heat, friction, when you rub it against that matchbook. However, if you lowered how much energy was needed, maybe you just had to barely touch the matchstick to the matchbook, it's more likely to happen. It's more likely to start. If it requires a lot of energy, a lot of back and forth, less likely to start. I found this diagram that I really liked with the frogs jumping over the wall. If this much energy is needed for reactant frogs to become product frogs, some of them might not have enough energy to jump over that barrier and become a product. But if an enzyme is present and they lower that barrier, it's much more likely that frogs, reactant frogs, can become product frogs. So by having enzymes around, it's much more likely for reactions to occur. Now, as I said before, enzymes are proteins. In most textbooks, they're usually drawn as a big blue or big purple blob. I don't know why, but they usually are. Shape-wise, they have an active site, and this is where some type of substrate, some type of reactant, something that's going to get changed, that's where that is going to react with. So the substrate is always going to sit in the active site, which makes sense. It's an active site. Something's going to happen there. Enzymes are very specific for what substrate can fit in their active site. Now, once a substrate fits into an active site and they're specific, they do form a more perfect fit. So it's like once they attach, they've become a more perfect fit. So this substrate, it's similar to the enzyme's active site, so it will fit. And then if you notice, this was curved, this was straight. Once it fits, then they form a nice perfect fit. This is showing an example as well of an enzyme working with a substrate. So here's our enzyme, big blue blob. This is our substrate. The substrate will fit, then it will make a more perfect fit. And the job of this particular enzyme, you don't have to know what this enzyme is, but it's called fructose 1,6-biphosphate aldolase. And no, 99% of enzymes end in ase, so then you know it's an enzyme. There's a few that don't, but most of them end in ase, so you know you're working with an enzyme. And a lot of enzymes are named for the substrate that fits there. So this enzyme's name, fructose 1,6-biphosphate aldolase, and that's because the substrate is fructose 1,6-biphosphate. So once that fructose molecule fits into the active site of this particular enzyme, the job of this enzyme, it's going to split this fructose molecule into these two smaller molecules, these two small products. However, that enzyme did its job. It split the fructose molecule into two, and then it goes right back to being empty and trying to find another fructose molecule to break up. So enzymes are not used up or changed. They're constantly recycled. This enzyme's job is to keep finding fructose, breaking them up, and then going right back and doing another one. They are specific for one substrate. The only thing that can fit in this active site is specifically this one fructose molecule. And their drive is to drive a reaction one way. It's a catalyst, and in this case, its job is to drive a reaction. It's a catabolic reaction because it's breaking something down. But it's only driving a reaction one way. This enzyme cannot take these two molecules and go in reverse and build a fructose. It can only break things down. There'd be another enzyme needed to put them back together. Now, there are certain things that can change how well enzymes work. One of them is temperature. And these are going to be some of my next slides. But temperature, pH, how much enzyme there is present versus how much substrate is present, and if there are any inhibitors present. So next slides. So some factors that affect that enzyme. This is showing that if you increase a temperature, how well that enzyme works, its activity will increase up to a point. Enzymes do have temperatures that they work best at. And enzymes that work best in our human body and for most bacteria that we work with in our lab, the enzymes work best at about around body temperature, around that 37 degrees Celsius. Enzymes also have an optimal pH, which is usually our body. pH, so right around 7, same thing for bacteria. If you change the pH, if you change the temperature, enzymes are not going to work as well. If you increase how much substrate is present, so if I go back, if I increase the amount of the substrate, if I increase how much fructose there is, this enzyme activity will increase up to a point. At some point, it doesn't matter how much fructose is around, there's only so much enzyme to break that fructose molecule down. This is just showing examples of two different prokaryotic cells that have two different optimal enzyme temperatures. This has an optimal enzyme temperature closer to body temperature, where if you've got a prokaryotic organism that lives only in hot springs, its optimal enzyme temperature is going to be much higher, 70 plus degrees Celsius. So different organisms can have different optimal enzyme temperatures. And then the last thing that can affect enzymes and how well they work is if there's any inhibitors around. And the inhibitors are grouped in either to a competitive inhibitor or a non-competitive inhibitor. So big blue blob, here's my enzyme. And the substrate that's supposed to fit in the enzyme, it's supposed to fit in the active site right here. However, there are some molecules called inhibitors that they're not the exact same shape as the substrate because they're not that substrate, but they can fit in the active site. And if this inhibitor is sitting here in the active site of this enzyme, well, the substrate can't fit there anymore. So it's called competitive inhibition is because these two things are competing to attach to the active site. So if the inhibitor beats out the substrate, that substrate can't attach and it can't get broken down or built up or whatever it's supposed to happen. You can't make the product from that substrate. The reverse, instead of having a competitive inhibition, you can have non-competitive inhibition. So again, same enzyme, same substrate, big blue blob. This substrate's supposed to fit here. However, this particular inhibitor, this little green circle, is present and it attaches to the enzyme in a different spot. It's not competing for the active site. So it's not competing with the substrate to fit in the active site like right here. That's why it's called non-competitive. But when this little molecule attaches to the enzyme changes shape just a little bit. And on this one, it changes shape by having this little hook. And because this hook is now present, because the inhibitor made that happen, because this little hook is present, the substrate can't fit anymore. And if the substrate can't fit, no product is formed. So it's still an inhibitor. It's still going to stop product from being formed. The only difference is the inhibitor is not competing for the active site. Now, what can happen sometimes is when a cell wants to make something, sometimes that final product can become an inhibitor. For example, this is showing a pathway, which means this orange circle is what the cell has. But the cell's like, well, I don't need orange circles. I ultimately need to make little green ovals, whatever that end product is. So it uses a number of enzymes to ultimately make the little green oval. So this first enzyme can take little orange circles and it will spit out little yellow triangles. This next enzyme is like, great, I love little yellow triangles. I will take it and I will spit out little red squares. And this last enzyme's like, awesome, I love little red squares, and I will take it and I will make little green ovals, which is exactly what the cell needs. However, the cell only wants to make what it wants to make, and then it wants to stop. So why make more end product if you don't need it? I only need a little bit of this. So that end product can actually become an inhibitor, and it can then go back and attach to that first enzyme. It's a non-competitive inhibitor. It attaches to the enzyme, and because it attached to the enzyme, that enzyme changed shape just enough that that little orange circle can't fit anymore. And so the cell will stop making that end product. And so again, the cell wants to make the end product until it's got enough, and then the end product becomes an inhibitor. Otherwise, it's a lot of energy to be making products that you don't need. And we're going to end right there because we're going to start with carbohydrate catabolism in lecture. And again, carbohydrate catabolism, it's a catabolic process, so we're going to be breaking things down, specifically carbohydrates, more specifically the glucose molecule.